Hey everyone, can you hear me? Good morning. Can you guys hear me over in the back okay? All right, I see some thumbs up, so I think that's a good sign. We did ask ARM to make this earlier at 6:45, so I apologize about the late start. So thank you for being here today. My name is Anshul Mangal. I'm the president of Project Farma and Precision Advance, and it's a pleasure to moderate this panel this morning. This topic is near and dear to all of us at Precision Medicine Group. Our interdisciplinary teams, including Precision for Medicine, Project Farma, and Precision Value and Health, have been advancing clinical and drug development, manufacturing, and commercialization for the regenerative medicine sector. Having been a partner to over 100 advanced therapy organizations, we understand the complexities to bring a cell and gene therapy to market. So in our panel today, we'll be hearing from our experts on these various complexities. They will give us an update on the respective companies, where they are at in their development process, and perspective on the challenges that lie ahead for the sector. So I'll briefly introduce the panelists, and then we'll get right to it. First, we have Stewart Abbot, Chief Scientific Officer of Turnstone Biologics, Andrew Knudten, Chief Operating Officer of Jaguar Gene Therapy, Laura Lande-Diner, Chief Business Officer of Satellite Bio, Robert Peters, Chief Scientific Officer of Ensoma, and Deb Phippard, Chief Scientific Officer for Precision for Medicine. So thank you guys for joining us. And Stewart, I want to start with you and hear more about Turnstone from you, its pipeline, and its mission to deliver breakthrough cancer immunotherapies.
Certainly, and thanks for having me the opportunity to be on the panel this morning. Oh, it's just, Mike's just kicked in. So Turnstone is a clinical stage biotechnology company. We focus on developing therapies for solid tumors, ideally with a curative intent. Our platforms are based on two different technologies. One is a virotherapy based on vaccinia, oncolytic virus, and the other is a tumor-infiltrating lymphocyte, or TIL platform. We have a long-standing interest in our virotherapy that's actually in clinical development at the moment, going through a dose escalation, phase 1-2, and we have just started a key collaboration with the Moffitt Cancer Center to develop selected TIL therapy in melanoma, and later in the year we hope to have a number of other indications starting for our TIL therapies. The key differentiation for the company is actually TIL therapy is that we're working on a selected TIL product. This is a tumor-infiltrating lymphocyte where we're selecting the tumor-reactive T cells out of the bulk TIL product with the intent to increase the potency of the TIL product in the solid tumors. And so we'll talk a little bit more about that later, but thanks for the opportunity to be on the panel.
Thanks, Stewart. Andrew would love to learn more about Jaguar, its pipeline, and where it currently stands on accelerating medicines for patients suffering from severe genetic diseases.
Great. Thanks, Anshul, and thanks for having us all here today. As you stated, Jaguar was built to accelerate gene therapies to patients, and we went about this in a way where we focused significant resources and attention in developing a very robust and strong CMC platform that would ultimately provide the purest and most potent gene therapies to our patients. And so we have 3 programs that are all at the preclinical stage right now, ranging from the rare disease space to more common disease. Galactosemia, the first one, also autism spectrum disorder, SHANK3 mutation, and then rounding things out with the big one is type 1 diabetes.
Thanks, Andrew. Debs, would love to hear from you more about Precision and the work that you're doing to advance cell and gene therapies.
Yeah, good morning. So Precision for Medicine is a full-service support organization. We do everything from late-stage preclinical, so looking at immunogenicity assays, all the way through to design of clinical studies, very heavily in the cell and gene therapy space. Under our Precision Advance banner. So the whole, how are we going to run the study, how are we going to do the translational assays, putting together all that immunogenicity work. And of course our Project Farma division, I need more coffee. They're very heavily involved in thinking about CDMO, how are you going to get all those products manufactured, how are you going to get them to market. So soup to nuts, anything that you could think of, Precision's there for you.
Thanks, Debs. Laura, would love to learn a little bit more from you about Satellite Bio and your proprietary satellite adaptive tissue platform.
Absolutely. Hi, everyone. So Satellite is pioneering a new therapeutic modality that we like to call tissue therapeutics, which effectively enables the broad use of solid organ cells as therapies. I don't need to tell this audience how effective cell therapies have been in the past, but so far, this has been mostly leveraged for immune cells. And the reason for that is that when a cell doesn't normally live in the blood, it's not going to survive and function for long when delivered by infusion. So our SAT platform effectively provides an in vivo support system and a delivery system to enable solid organ cells, such as liver cells or pancreas cells, to survive and thrive in the host and deliver a therapeutic function. And the value of this, I think, is mostly in the context of diseases that have been elusive so far, where perhaps targeting one aspect of the disease is not going to be sufficient, and this therapeutic approach enables us to take advantage of the full breadth of functions that the whole cell has and use it for therapeutic purposes.
Thanks, Laura. Robert, last but not least, love to hear more about Ensoma developing one-time in vivo treatments that precisely engineer any of all blood and immune cells to cure disease from within. So can you tell us more about Ensoma?
Sure. Thanks, and thanks for having me on the panel. So we at Ensoma are focused on in vivo cellular engineering. We use a virus-like particle that can deliver 36 kb of cargo to the nucleus of any hematopoietic cell. And so we've now shown whether you're looking at mature cells like T cells, NK cells, or to hematopoietic stem cells to get the durable effect from their progeny. We're able to put all this together to incorporate any gene engineering tools such that we can create unicellular or even multicellular medicines depending on the application. For example, whether it ranges from genetic diseases with a single gene defect to more complex combination therapies, say a CAR-T, CAR-NK combination in oncology.
Thanks, Robert. So the first topic I want to turn to are the roadblocks kind of our industry has faced this year. And I really want to focus on, how to mitigate these roadblocks for the rest of this year and beyond. So Andrew, I want to start with you. Over the past 2 to 5 years, the cell and gene therapy sector has flourished with significant investor confidence. The recent downturn in the public markets has definitely influenced this confidence, 2022. But really my question is for beyond 2022, do you think the current state that we're in will continue, and what will be the outlook for the future years?
Yeah, it's a complex question, and candidly, there are many companies out there that just went out and licensed a construct and expected to be in the clinic tomorrow, and that's sort of what we're starting to see as this field evolves, to change things and to start to, I guess, move the needle in the positive direction. Is we need wins, right? We need wins as an overall field, and it starts from a CMC perspective, just providing that robust analytical process and really characterizing and understanding your process and your product really well. Once you start doing that, you start to get to the point where you're meeting and exceeding regulators' expectations. And that's where we need to be to achieve these wins. If you are in that environment, you're going to have fewer issues, you're going to have fewer holds, and you'll have more clinical success. And so, I think, to me, that's where the industry needs to go. We need to get more wins, and then the money will follow. So I think the investors and the money and that sentiment will change over time.
Laura, Robert, from a therapeutic developer perspective, how have you weathered the storm, and has your strategy shifted at all?
Yeah, maybe I'll start. So we were lucky at Satellite to launch earlier in April with over $100 million Series A raise in cumulative funding, and so right before the market downturn. So we are fortunate in that way, but certainly the situation has impacted our strategy, I would say, in 2 primary ways. First of all, it forced us to focus our efforts and our money into the key goals of a company this stage. And oftentimes, platform companies can get tempted and distracted and do more than one thing or more than what's critical, and that's a problem. And so we have been much more focused. The second thing, which I actually think is an opportunity, is that now we are spending more time and acting on partnerships of the kind that I like to call synergistic partnerships, meaning partnerships where both parties bring technologies and capabilities to the table, and both parties also stand to win by the joint effort, right? And I think the markets and the sort of the horizons in terms of financial resources has forced us as a field to think again about these partnerships, and ultimately this can be a huge win for all of us.
Yeah.
Robert, do you agree?
Yeah, no, absolutely. Actually, it's funny, I noted in the latest ARM summary of this sector, the one quote, I believe it was the direct quote, is the strongest punishment has been doled out for early stage cell and gene therapy. Woot, woot, yeah. But, so lucky us, yeah. But that said, you know, some things, ultimately, whether it's market forces or whether it's, you know, internal corporate priorities at larger companies, it does get back down to really three things, right? So you want to really drive a strong scientific foundation for the work you do. You need to apply that in a way that will bring value to patients and their families, and you need to be able to convey that to your key stakeholders. And you know, if you're able to keep doing that, I think on some level, you know, good science will end up, you know, doing very well for itself. I should also say it's worth, while you drive your platform forward, it's important to be, you know, always looking for those opportunities. Where else can you go, and what other value can you generate? I know in our case, you know, we started with really focusing a lot on reproducing all of our founders' work, and I should say it's always nice when you can reproduce your founders' work, but not always a given, but really taking that focus initially more in genetic diseases, but then we've continued to apply that to, say, engineering the immune system to actually open, you know, opportunities in oncology. And I think, you know, it's that combination of just, you know, focusing on those fundamentals and driving things forward. I think things will put us in a good place.
Stewart, what's your perspective? Do you guys, is it the strategy that, you know, that folks have already laid out here, is that kind of what you guys are following as well?
Yeah, I think by and large, yes. You know, one of my, a good colleague of mine once told me, good clinical data solves all. And so at the end of the day, what we're really pushing toward is to get as many of our clinical studies started as possible, as efficiently as possible.
With the best design possible.
With the best design possible, yes, because you can take a good drug and put it into poor design and not get a good outcome. So I think it's critically important to think about all of the complexity of this. But really, we're not being valued as an industry now on our non-clinical data. By and large, we're being valued on our clinical data, and I think that's largely appropriate. And so getting that clinical data efficiently and effectively is our key goal. We have studies starting, as mentioned, in melanoma. We are starting studies in a range of other indications as well, ideally with the correct clinical design to get the best outcomes as early as possible. Now, there is an element where we've been a victim of our own success, and so there's an expectation with some cell and gene therapies that we have success straight out of the gate. You know, first patient, second patient, third patient, they're going to be having miraculous recoveries, potentially cures. I think as an industry, we will hopefully evolve beyond that and realize that, you know, this is a complex modality. It's, you know, yes, it offers, cell and gene therapies offer an incredible amount of hope to treat complex diseases, but they still have the same challenges as small molecules. And as you're going through dose escalation studies and the like, you may not see success instantly, but with the right clinical design, success hopefully will come. So we're approaching this from a perspective of a number of different indications with hopefully the right clinical design.
Robert, do you think this will be the new normal for cell and gene therapies moving forward?
Well, you know, like, you know, just building what everyone else is saying, I think this is a moment in time, right? And and we have seen clinical successes, and even right in the last, you know, few months, a few approvals, you know, of curative therapies. And so I really think that ultimately that'll turn things around and, you know, drive more interest back into the industry.
Thanks. Stewart, jumping back to you, in addition to the markets being a challenge, obviously there's been tons of regulatory roadblocks that have been a challenge as well, disappointing study results, safety concerns for patients. How do you think this impacts the sector, and how do we overcome that?
Well, it has had some negative impact for a number of sponsors. I think one of the more interesting things, or more interesting statistics I heard at one point was that more than half of the investigational new drug applications that went in over the last 12 months were actually put on hold. And that was unusual. I mean, certainly in the past, if you look back over the past 5 years, that was an unusually high percentage. So I think we have to ask the question, why are so many new drug applications going on hold? And that's a multifactorial, multifaceted problem. The agency's had quite a lot on its hands since 2020. Little pandemic going on that they've had to deal with. Quite a lot of, we've seen, and I've seen this in a couple of different companies now, we've seen quite a lot of turnover within the US FDA, where folks that have had decade plus of experience in cell and gene therapy have moved on to other roles, and that means there are a number of new folks coming along. They're experienced, but only to a point in time, and so there's a bit of education that goes on there. And I think the key thing that a lot of sponsors perhaps don't realize is that we are somewhat a victim of our own success. We've had a lot of clinical trials that have been accepted and developed over the last few years. That's given, it's presented the opportunity for a lot of information to flow into the regulatory agencies, and they're assimilating that. And they are now in a position to ask quite relevant questions of sponsors. And I think we're seeing that being fed back to us in the form of advice and recommendations. The recommendations from the agency are not coming out of the blue. They're coming out of the massive amount of data that they have from the clinical
the bar's
studies.
going higher now?
I think the bar's going higher. Well, I shouldn't say the bar's going. Yeah, the bar does appear to be going higher, but I think it's really the knowledge level is going higher, so that the bar can be maybe appropriately presented to us. And our not just Turnstone, but I think the industry's solution to this is really to pay careful attention to the guidances. The agency's not in the habit of surprising us.
Yep.
They do provide guidances. Sometimes they're a little behind the time, but they do provide guidances. And if you look at those carefully, digest, and ask the right questions, then I think you can get ahead of some of these issues.
Yeah.
Andrew, what's your experience been like communicating with the regulatory folks, and has your communication changed from the last 2 years to what it is today?
I think it's continued to evolve, and I think what Stewart said is spot on. I think the expectations have always been there, but I think in prior years, companies would go to the agency asking the questions: Is this good enough? Mm-hmm. Would you take this? Would this be okay? And that's not the case anymore. And you look at I don't know how many hundreds of AAVs that have gone into, and we'll speak about the gene therapy side, that have gone into the regulators. There's a great database of information to be able to set expectations. And so, and we've got great forums for dialogue. We were just in an Interact meeting recently, and those are great forums where you can actually have a true dialogue and say, we are doing this, and this. We anticipate this will meet and exceed your expectations. If those are the questions that you're asking, and the feedback that you're trying to gain from these regulatory dialogues, then we will move things forward in a positive and successful way. So yeah, absolutely. I think the bar was always there, but now that you're really being held to it.
Yeah, so a little bit of what Stewart said, victim of our own success. Debs, how does an early phase company anticipate and mitigate some of these regulatory hurdles if they don't have an Andrew Knudten, that's shepherding them through the process?
You're throwing that question at me? Yeah, thanks for that. It's really around planning. It's really engaging early, asking the questions and being able to plan ahead of those questions. The last thing you want is to put a data package together, go to the FDA and have them say, oh, we need to see this, this, and this. A lot of the work that we're doing is in the immunogenicity space. So we're looking at a companion diagnostic in parallel with a BLA in the case of gene therapy. So you've got to keep all those things going in tandem. You're dealing with regulatory agencies where you've got CDRH, you've got CBER. They may not be talking to each other as much as you would like. You're potentially in the middle of that, trying to negotiate with two different agencies. So to us, it's very much about think about it, get in early, plan as much as you can. And I would say this is constantly evolving. I was just at the WRIB meeting last week. Precision does a lot of translational work in the cell therapy space. So we're looking at distribution and persistence. When we get high enough cell numbers, we're looking at, are they activated? Are they becoming senescent? The FDA is now looking at regulating those assays. And it's been very much a free-for-all. You know, you put a flow assay together, we say it's great. Now they're saying, well, how are you going to regulate that? How are you going to control that? So for us, the bar is constantly moving, I think in a good way. Nobody wants to put bad data in front of a regulatory agency. Nobody wants to risk patient safety, but the goalposts move constantly. And when you're working on an assay, we're constantly looking at, is the guidance changing? Are we getting more white papers? We've had to pivot midstream. Classic example, we were working on a clear assay. We're ready to go. They're like, no, you've got to regulate that to CLSI guidelines. So without losing timelines, you've got to pivot. So I think we're in a constantly evolving field in a good way. But that's why I was asking to you. I feel like the bar is definitely moving up. And that's a good thing for the field, but it costs more money, takes more planning, takes more time. Nobody wants to add more time to their clinical studies. So we have a lot of things to keep up with, yeah.
Robert, how have you mitigated hurdles as Ensoma with the regulatory bodies?
Yeah, well, so, you know, we're fairly early days,
Yep.
but, you know, making sure that we're setting a good foundation for those future conversations, whether it's for planning what we're going to do for, you know, clinical design or for manufacturing, starting early for getting, you know, characterization assays in place so that ultimately, you know, comparability doesn't become an issue way down the line as you move forward into the clinic.
Laura, any final thoughts?
Yes, I mean, it's been said, but I think the importance of early interactions, especially for advanced therapies and new modalities, is extremely important. We at Satellite had an Interact meeting last year for our lead product, and to have the opportunity to ask the right questions about manufacturing, population targets, and the product itself has been extremely helpful.
Thank you. So in addition to market pressures, regulatory, I think pricing continues to be a challenge. I want to ask a kind of a different question. How do we balance the cost of these drugs with the current one-year budget system that our healthcare system is predicated upon? Andrew, would love to hear your thoughts on that.
I think this is where we can do so much better as a field in our approach. I mean, if you want to decrease the cost of healthcare, you just can't go tell biopharma companies just charge less, right? And we've been in that sort of whipsaw and spiral where eventually that's going to impact, you know, the sort of cost of quality, and that ripples through to patients as well. You have to have a time horizon beyond 1 year. We all run businesses, and you set your MPV on a 10-year horizon roughly, but you know, can't we just get to maybe 3 to 5 years from a healthcare perspective? A 1-year budget doesn't allow you to really compare the benefits of something that you're going to see those transformative or curative benefits over a longer period of time versus you know, treating symptoms on a daily or weekly basis. And so I think it's hard. It's difficult, but we have to keep trying and start to move the needle towards a longer time horizon.
Thanks, Andrew. So would love to move into some of the new technologies that are making waves, that have made waves in 2022 and are poised for growth in 2023. So I'll throw a very general question out to the panel because I would love to hear you guys' thoughts. Is what would you say are some of the top technologies that are poised to disrupt advanced medicines? And Andrew, would love to start with you again on that one.
Yeah, I'll just kick things off and turn it over to everybody else here. I think it's a story that's still being written. We're in chapter 1 right now as far as gene therapies are concerned. If you look at AAVs, chapter 2 will move into, you know, engineered capsids, and chapter 3 really on the non-virals. And every one of these modalities has a place. It's, you know, and what I try to coach my group and coach the folks that we're working with, even the bankers and investors, is one thing doesn't have to be bad for another to be good. There's a place for many of these therapies across the board. There's great companies out there like a Saligen and Cove Therapeutics that are doing great non-viral work. There's great companies like Capsida and others that are doing work on the engineered capsid side. There's a lot of great companies that are out there where there's the most data at the FDA right now around AAVs, and I think they all have a place. So,
Stewart, Robert, love your thoughts here.
Surely, I mean, there are a phenomenal number of technologies that have developed over the last few years for manufacturing, for, you know, vector design, for engineering, etc. But I think the thing that I'm probably most excited about at the moment is actually trying to solve a problem that we've had as a field for quite some time. And the problem that we've had, or one that's plagued me certainly, is the sort of lack of non-clinical models, predictive non-clinical models for safety and efficacy of complex therapies. There aren't many good animal models. There aren't so great in vitro models, albeit they can be helpful. And so when we're going into the clinic, we're going in a little bit blind. And so as we've gathered information, I mentioned this vast amount of information that's flowed into the field over the last few years, I think we have an opportunity now to think about that sort of bedside to bench transition, the translational aspects back from what we're learning in carefully designed clinical studies with good translational endpoints, good investigational endpoints, and start to use some more complex computational systems, and AI is the wonderful buzzword, but there's machine learning's been around for years now, and start to use some of those machine learning technologies to interrogate the vast amount of data that we have from the clinical studies and hopefully back translate that into better drug design as we go forward.
So is that a plea for companies to share more of their early data? You think that would help the field? I mean, we all kind of keep it fairly tight, right?
Yeah, it'd be nice to be pre-competitive, but I mean, yes. I think there's, just types of four always discuss sort of pre-competitive information and whether the different regulatory agencies as a melting pot for that information can share more of it. I think there are always challenges with that, but we keep on having to try. And some of this is through publication. I think that one of the things that has happened more recently is there's a lot more supplemental data coming out in publications and a lot more mineable supplemental data coming out in publications. Now, there's always questions about the quality of the metadata, et cetera, but we're starting to see a bit more of that we and others can mine into, even if it's not our own data.
Everybody, please email me if you want Stewart's data. Happy to bring it to you after this meeting.
Certainly.
So, Robert, what are your thoughts?
Yeah, so when I think of what's going to be really disruptive, it's maybe not a single technology, but certainly when I look at engineering the hematopoietic system, there's more and more advances have been made in ex vivo, but I think there's more and more companies now looking to translate that into in vivo by one means or another, and really the thought that we'd be able to take those learnings and all those curative effects, but then translate them into a way we could really get to a much broader audience, a much larger patient population that could benefit from those advances, I think is going to be really exciting to watch.
Laura, obviously, what Satellite's doing is fairly nuanced. How do you see tissue engineering playing a role moving forward in the regenerative medicine sector?
Yeah, so absolutely super excited and a big fan of what we are doing, and I think finally enabling other cells to also be used as therapies has tremendous potential, especially for complex elusive diseases. I think on the flip side, what excites me is the use of patient data, as Stewart was saying, for certification. I think the diseases that are complex are very nuanced, and many times because of poor study design, but also because of poor patient selection, many drugs that could have succeeded don't. And I think today we are in a position to develop really amazing drugs and at the same time really understand patients so that we can match those drugs to patients much more efficiently.
Debs, what tech are you most excited about?
Ooh. So it's really difficult to pick one. So the fabulous thing about working for a service organization, which by the way, when I was a Ph.D. student, everybody said, don't do that. But it is so fantastic to be able to see all these different modalities, to think about the best way to test them. Obviously, clinical design, the last thing you want to do is disadvantage a great new therapy. There's space for so many of them because they're really targeting different problems, different concepts. I get really excited about the CRISPR technology because I think it is so flexible. There is so much you can do with being able to take cells, engineer them, give them back. I think there's a lot of safety challenges with that we're going to have to work through downstream. I get very excited about all the new vector technology, the whole concept of how you can target it, tissue specificity. Do we really have to worry about immunogenicity? If you treat neonates, what happens? Do you keep expression levels as those patients move into adulthood? How do you think about retreating? Basically, I'm just excited about the field. I'm not going to pick one. I don't think that's fair. We can talk about it at coffee, but this field is so expanding. There is so much opportunity. And as we said earlier, nothing is going to help us like the early successes. It will bring more money back to the field. There's just a lot there for patients.
Thanks, Jeff. And I'm happy that you went into a service-based organization. Thank you for that decision.
We can talk about that at coffee as well.
Yeah. Laura, what about non-viral delivery methods? Do you think those will replace viral delivery methods, or is it there's room for everyone?
I think, as Deb was saying, there's room for everyone. I think there are different delivery methods are tailored to the entity that they need to deliver and the reason for the delivery. I think at Satellite we have a delivery system. In some ways you could think about it that way for cells, right? That's not exclusive of delivery systems for genes of different sizes and so on. So I think absolutely there's room for everyone, and the need definitely has not been met.
Stewart, Robert, any final thoughts on non-viral versus viral?
Yeah, I think when a lot of times people think of non-viral, they think mostly lipid nanoparticles. But I think even what we've been looking at are these virus-like particles where you've taken the viral vector but removed all the viral genome. But you can still leverage the hundreds of millions of years of evolution to have a viral vector deliver cargo directly to a nucleus. And so again, I think there's a lot of different ways to approach different problems. But that's something that we think is going to be really interesting to watch.
Stewart, what do you think?
Yeah, I mean, viral vectors will be with us for a while. Well, they have been with us for millions of years. They will be with us for a while, but we're always going to strive towards more simplicity. And so if you can have a vector system that you can make in 2 weeks rather than 2 months, that has the same operational efficiency as the vector that takes 2 months to make, you're going to go with the vector that takes 2 weeks. We're not quite there yet, but we're moving in that direction.
Yeah, and don't give me empty capsid. That's really annoying.
So we've spoken a lot about gene therapies. Obviously, there's been a lot of innovation in cell therapies as well. Specifically, allogeneic cell therapies have seen a great deal of rise this past year, although many of them are facing durability issues. Will these therapies overcome and offer a more efficient solution to autologous cell therapy, Stewart?
Oh, I'm conflicted on this one. Some of the audience members will know I've spent most of my career working on a mixture of allogeneic and autologous therapy, so I love them both equally. No, they're.
Do you have a favorite child, or are they both you love them both?
I also have 2 children. I constantly ask which one's the favorite child, and I refuse to answer that. But no, I think. Look, again, it's sort of what's fit for purpose, and so. If you think about autologous cell therapy versus allogeneic, they have different opportunities, they have very different challenges. I think the key challenge for an allogeneic cell therapy and a gene-modified allogeneic cell therapy is integrating the immunology. And I'm talking predominantly around the use of those cell therapies in immuno-oncology. So there is a complexity to the immunology for an allogeneic therapy that we have yet to fully solve, albeit there are a number of groups that are making great strides in that direction. And a lot of that is understanding not only the effect of the sort of drug on the body, but the effect of the body on the drug, the persistence, immunogenicity, etc. Now, on the flip side, when you're talking about autologous therapy, you have a system that is perfectly integratable back into the patient that it came out of. However, you do have some manufacturing challenges, and I think there we have to solve for the complexity of the manufacturing. So if we solve all the complex immunology problems for allogeneic therapy, they will be successful in certain indications. And if we solve the manufacturing challenges for autologous therapy, then I think there's certainly a room to be effective. And obviously, we're working on an autologous patient-specific tumor-infiltrating lymphocyte, and I think that's really harnessing the key attributes of that cell to react to a broad range of tumor antigens and then to persist thereafter in the patient.
Do you think there's tolerance for the cost of autologous therapies going forward?
Yes. I mean, the simple answer is yes. I mean, and the economics is incredibly complex problem, but it really comes down to what is the efficacy. Yeah. I think if you have if you have a product that costs a few $100,000 to make, and you have efficacy that only lasts for a few months, I think that's challenging. Mm-hmm. If you have a product, whether it's a gene therapy, autologous therapy, or allogeneic therapy,
it's much more durable.
That costs a few $100,000 to make, but you have curative outcome, then yes, I think it's certainly possible. So it really comes down to what is the return on that investment, and do we have durable outcomes?
But it's more than the cost, though. I mean, that's the challenge. I mean, it's don't get me wrong, it's great to see curative therapies, but the manufacturing complexity does limit the number of people that you can reach to those therapies. And so it's great to see those moving forward and looking to build on that. I know allo is one way to try and go about that. But certainly I'm optimistic. It's early days, but for the future when we can have an off-the-shelf therapy like, that you can dose a patient with this off-the-shelf therapy to reprogram the patient's own cells and really cure from within. And that's something that I'm really excited for in the future.
I'm glad you guys brought up manufacturing because that's a topic that's near and dear to my heart, and I think it's one of the last topics that we're going to cover today. The internal versus external thing has been debated ad nauseum, I feel like, and I think previously the sentiment was scale up internally, scale up internally. You control your fate if you scale up internally, and a lot of that was driven by lack of innovation at external manufacturers. Andrew, do you think that's shifted at all? Where do you think that paradigm lies today?
I think there's a lot of capacity out there, but I don't, I think there's just not a lot of competent capacity out there in the space. And it doesn't have to be.
That was definitely a dig at Anshul.
Tell us how you really feel, Andrew.
Move a little.
No, but I think it's part of the evolution of understanding what's important in the advanced medicine space. I think people forget that you do have to do the blocking and the tackling and just be responsive, do all the things that a good CDMO partner can do. It's nice when you've got a supercharged cell line or some technical hook, but just deliver, be responsive, be rapid in turnarounds, because we all want our individual medicines to proceed and accelerate to the clinic as soon as possible. And I say competency in a very simple way, just do few things well.
I think it's do your job, Anshul.
Yes, yeah. So and there are many companies now that are merging, that are setting themselves apart in the space. But I think you have to create that fallow space, and then eventually you fill it up with the competency over time, and we're seeing that now.
Robert, what's Ensoma's manufacturing strategy?
So, it's interesting to say because it is difficult to, as a small startup, to actually start with manufacturing from within, right? But certainly manufacturing is such a critical part of what you do. What we chose to do is really focus on manufacturing from the get-go, developing internal expertise. We brought in a chief technology officer to build these capabilities, actually before they brought me in. So, right, they definitely bringing in those core competencies. And what that allowed us also to do is really focus more on internal research manufacturing as well as process development to then partner with really good CDMOs to actually drive the manufacturing process forward. But what this also has been allowing us to do is to actually really make sure that those manufacturing people are working hand in hand with our platform, our research platform advances, and so to make sure that as we plan for the future that we're optimizing for manufacturing the processes that are evolving within our platform as we maybe change directions here and there. And so that's been a critical component. And then just setting ourselves up for future success. We mentioned earlier, making sure that we're building also the analytical tools within our company to really understand our own products so that we can really give ourselves a good basis for the future and being able to do characterization assays that will go across all our products and set us up for comparability down the road, et cetera.
So can I just jump in there, because
Yeah,
that
please.
is such an interesting point, because you mentioned analytical assays, and that to me is a spot that we often miss early on when we're thinking about manufacturing. So if you're in a situation where you have to make a companion diagnostic, if you're using like a luciferase AAV reporter construct, you need 3 batches of that to close to GMP grade to be able to go to the agency with your CDX. I cannot tell you how many clients I've had where I mention this and they all fall over with, we've got no time to do that. We've got no manufacturing capacity to do that. So being able to do that early on, you don't need massive batches, but it's the level of quality that you have to put together to satisfy the CDRHs of the world that your assay is robust. So thinking about analytical reagents, it's not just the drug product. Everything has to go in parallel. I think we're a lot more aware of it now. It's fantastic that you mention it. But I put that plea out there because, it can be a really nasty surprise.
Completely agree, and I think having some of that ownership internally from a non-GMP perspective, and I would even venture to say going full scale non-GMP allows you that capability to characterize, and having those analytics in parallel or even slightly ahead of your process, as you stated, is incredibly valuable.
It's really straightforward. If you can't measure your system, you can't control your system. That's right.
Yeah, and we need a bit longer than 3 months. I'm just saying, everybody.
That's actually one of the challenges that we face for manufacturing. We have outsourced our manufacturing for our early stage clinical development. We work with a number of hopefully quite competent CDMOs. But I'll take your point. But I think one of the challenges is, we are working with external CDMOs, but we're also thinking to the future. What does success state look like? And one of the biggest challenges is because there's such a long lead time on the implementation of current manufacturing technologies, you actually have to start, potentially start investing before you have the clinical data to justify that, because of the long lead time. So I actually think there's a gap in the market for being able to transition more quickly into in-house manufacturing, whether that's modular systems, however that comes about, but not having to take a year plus to bring that capability in-house, and not having to pre-invest at a stage where it can be painful. It's much easier to invest in manufacturing technologies after you have some clinical data to justify that, but because of the lead times, oftentimes you have to do that ahead of, and it's a little uncomfortable.
Yeah, for us, we absolutely, appreciating the experience of others, we made the decision to invest in manufacturing from day one. As you mentioned, we have a CTO from the beginning of the company, and we are investing alongside R&D. We are investing on manufacturing alongside R&D because we realize that it's not enough that the technology works in mice or also in patients. You need to be able to scale it and expand it in a way that makes financial sense as well. So we went with the camp of building our own manufacturing facility and owning that 100%.
Seems like it's kind of a chicken and egg problem. You need good clinical data to raise the capital you need to invest in internal manufacturing, but you need internal manufacturing to get the good the drug product.
Maybe we need those ex vivo models, the animal models.
So I guess we're almost short on time, so I'm going to ask one final question to all of you. What are you guys excited most about for your respective company, for the sector, in the upcoming year? I think this year's been a challenging year. It's been an up and down year, but I still see there's a lot of hope for patients that are out there, and we need to provide that hope for the patients that are out there. So what are you guys excited about, and what are you guys hopeful for? Robert, start with you.
Yeah, I have to say, within Ensoma, I've been really pleased with the progress we've been making with the platform. You always get some, twists and turns along the way, but really driving that forward and opening greater opportunities is something that we've been making some nice progress in the last few months in particular, and really looking forward to that for the future. And as far as the industry, market pressures, aside, there really have been a lot of advances. We really just have had a number of recent drug approvals of new ex vivo therapies, both in US and Europe with, in vivo therapies for hemophilia. All these are moving forward. We're seeing clinical data, reading, you can't seem to go another month without a new CAR against this, that, or the other target that's really showing great progress in areas where there's just tremendous need. And so I think it's exciting. I'm really looking forward to the future and what we'll bring.
Debs?
Yeah, to reiterate, we're getting to see great clinical data. I think I'm excited to see all the new second, third stage vectors that are coming out. People are finding new vectors in rhesus monkeys. They're thinking about, can you truncate proteins so you can pack more into an AAV? How's that going to work? Can you think about more tissue specificity, promoters, vector capsids? So much that is happening in that field that I think is really exciting. I agree, viral vectors are not going to be going away for a while, but there's a lot to do that we can make them better. The flip side of that is I hear a lot of times, it would be fantastic if we had one immunogenicity assay. You could build a neutralizing antibody assay. You could use it for all viral vectors. I get that concept because building those assays is time-consuming, expensive. You've got to think about the cutoffs, your patient population. But I almost see the field going in the opposite way. We're getting more and more specific vectors, which to me really brings up the question of immunogenicity and what really matters. So I'd love to see more work on, what does pre-existing immunity mean in terms of efficacy? Sure. I think we're starting to work that out, but we've got quite a bit of way to go there, but lots to happen in the future.
Laura?
For me, it's the use of cells as therapies, right? Our platform, it's the combination of the use of cells as therapies that has expanded beyond immune cells, and more companies are developing iPSC or primary approaches to use other cells. And alongside that, we have this situation that is forcing people or encouraging people to partner for synergies. And I think that this complementarity can be extremely valuable for patients.
Yeah, I think I was on a recent panel with Jim Wilson. He says, I think one of the biggest trends he sees was consolidation. There's going to be more partnerships, there's going to be more consolidation and better portfolio construction going forward. So I think we're seeing a lot more of that, and I think that's going to be good for the industry. Stewart?
From a field perspective, I'm hoping that 2023 and 2024 will give us more insights into how to use gene therapy for polygenetic disease, not just monogenetic disease, and that's just from a field overall field perspective. Being a little parochial from a Turnstone perspective, 2023 will be a transformational year for us. We'll have the first of our tail data coming out. Really looking forward to that. I think it's exciting. The platforms, does have quote-unquote transformational potential, but we really have to prove that in the clinical data. And we'll continue to see the clinical picture for our virotherapy evolve. Beyond that, we're working with some great people, both inside the company and outside with different CDMOs and the like, and that's an exciting part of our work going forward. And then we have a large non-clinical development or non-clinical pipeline that will be making its way through into clinical development in the 2023-2024 timeframe. So lots of exciting things going on.
Thanks, Stuart. Andrew, any final thoughts?
I'm just excited that a lot of the technical improvements are focused on patient safety. And really, cost of goods, they all kind of correlate, right? But if you focus on the patient, you focus on patient safety, other things will fall in line as well too. So because we want to be in an environment where we can treat patients and that risk benefits, not this huge thing hovering out there saying, we'd like to treat them. However, is the risk benefit good enough opposite this viral vector or this next generation gene therapy? So keep focusing on the safety.
Thank you, everyone. Thank you to all of you for providing those great insights today. Really appreciate it. And thank you, everyone, for listening.
Thank you, Anshul.